case, we can tell in terms of robustness, sensitivity is a multi-way comparison. For other characteristics of the experiments, you can go to our application note 4 to find the detailed information. Now, first thing first, we want to work out the way of comparison. First thing is we want to have a good LC-MS separation. We try different salt concentration. We end up with 200 millimole ammonium acetate. Then we try different flow rates, 1 to 10 microliter per minute. It turns out 5 microliter per minute give us the best peak shape as well as the throughput. So for the later comparisons, we focus on 5 microliter per minute, 200 millimole ammonium acetate. Then we use the standard IgG antibody from NIST because that gives us exactly the charge states, the different glycophones, and also the concentration for our later head-to-head comparison. So at the bottom, you saw the typical charge distribution of the IgG from NIST. We focus on the more abundant glycophones. In this case, 5 of them. So you have 2 heavy chain. Each heavy chain has a FC N glycan. So we have these 5 different combinations of the glycophone. So we use a thermal software data deconvolution. We were able to quantify these 5 proteophones for our later quantitation. Now, first thing I want to mention is the dramatic increase in sensitivity of our M3 emitter at 5 microliter per minute compared to healthy source at 200 microliter per minute. You can see for both 50 nanogram or 200 nanogram injection, we got at least 20 to 30 for increase in sensitivity. Then people ask, what if you compare to other emitters with the same flow rate? So this is the answer to that question. Most of the time, people use healthy source. So we did 1 microgram IgG injection at 5 microliter per minute. We got 5-fold increase in sensitivity. If we use other type of emitter like picotip, nanotip, we got at least 2 to 3-fold increase in sensitivity. This didn't, we actually realize robustness is dramatically different. I will show you how our emitter can run 150 runs without interaction with good CV in the next couple of slides. We also did the titration and found out the linear range for our emitter. So limit of detection is about 10 nanogram. That means our emitter at microflow can dramatically increase the sensitivity. Now, robustness is also a very important characteristic if you run native MS. As I mentioned, people don't run native MS using nanoflow because the tip tends to clog right away or after several runs. So we give us a high bar. We ask the question, can we run at least 150 runs for one M3 emitter? If so, what's the CV? So here is the answer to that question. As I mentioned, we use 5 different glycophones for quantitation. The most abundant one, we got about 4% in CV across 150 runs. We look at other less abundant ones as well. So on average, the CV is 7%. This means our emitter is highly reproducible and highly robust. As you can imagine, 200 millimole ammonium acetate. In addition, because native MS solution is aqueous solution, it's very difficult to ionize the molecules from aqueous solution. So 150 runs is a very good number. we now actually further push this using a bigger ID emitter. So we'll have the data very soon. Now, the other side effect we found using our microflow M3 emitter actually is very important when you do native MS, because you want to maintain your protein in the unfold or native state so that you can preserve the protein-protein interactions, protein machineries.
Over the overall proteomics, the overall omics program that we have. And when we really think about the omics vertical market, we think about four programs. The precision medicine program.
Sorry, can I finish the slides, then we can discuss at the end? Okay. So, as I said, the native MS, we want to maintain the native state. This slide shows that for HESI source, the maximum or highest abundant charge state is at 29. If you move down to microflow using HESI or using our emitter, the maximum is 27. That means the protein probably are better in the native state. Because if the protein are unfold, you expose the surface, you tend to have higher charge states. So this is so-called side effect of our microflow MS platform, but it is very important. We reason that maybe it's due to the HESI source, high gas flow, as well as the heating. So some people ask us, they don't have microflow pump right now. They don't have microflow column. Either way, how can they use our M3 emitter for their native MS studies of IgGs? So here is one solution. If you have a microflow column, if you have a high flow pump, you can do pre-column flow splitting so that the flow is microflow. You go into your microflow column connected with our emitter. Or you don't want to do splitting pre-column, you could do post-column splitting. In that case, you use high flow column, high flow pump. You use a 1 to 40 splitting. In the end, you may end up with 1 to 10 microliter per minute. So this is one example of using the splitting platform. So you start with 200 microliter per minute. You spread 1 to 40. We end up with 5 microliter per minute into our M3 emitter, into our mass spectrometer. In parallel, we did a comparison with exactly the same amount of injection using high flow column into the mass spec. We saw 30 times increase in sensitivity. Again, this is considering the amount injected into mass spec, not the beginning amount. The beauty of doing this is you actually don't need to inject a lot of material into your mass spec, because if you run native MS, your high sort, your IgG tend to clog your ion cone, so that you have to do maintenance once, probably every couple months. On the other hand, if you do splitting, material can be diverted into UV spectrometer or for other type of studies. So in that case, you can have one stone, two birds in one experiment. Of course, we are also working on the high flow solution. Essentially, we want to provide a product. You come in with high flow column, high flow solution, you can still use our emitter. So we are working on that. Finally, let me summarize with our technology and maybe open up for question and answers. So because we have been working with thermal for quite some time, as soon as we officially launch our product in June at ASMS 2019, thermal announced we're gonna co-market to really push the biomarker disease disease biomarker validation. So most of you are scientists. We know it is relatively easy to discover biomarkers. You can maybe identify several hundred biomarkers, but validation is hard. You need to run hundreds, if not thousands, of clinical samples in order to validate the biomarkers. In this case, you need throughput. On the other hand, most of the biomarkers were discovered by nanoflow. Whether they are reproducible, whether they are robust enough, that's the open question. So it will be nice if the discovery and the validation platforms were both robust and reproducible. And the other thing is a lot of validation was done by antibody validation. That will take a long time. So in light of that, Thermo worked with us. They put two of our application notes we joined that they developed on their website. So these are the bottom-up proteomics as well as targeted proteomics application notes. You can download them from their website. So our product right now is gaining momentum. So far, we have 68 early adopters, including Merck, Genentech, Regeneron, as well as CDC. So some customers are fairly happy with what they can do with our emitters. Dr. Wang from Regeneron, he was using our emitter for native studies of antibodies. He saw at least 3-fold increase in robustness, meaning he can run 3 more kinds of samples. His group actually was using our emitter for peptide as well as nucleic acid analysis. So Teresa from Stanford, we collaborate on her Bruker Message 2 instrument. So she saw at least 3-fold increase in sensitivity. Dr. Dai from Sanofi, as well as Bird from University of Lausanne, they were using our emitter for peptide analysis. They saw 3 to 5-fold increase in sensitivity. Let me summarize the characteristics of our emitters. Over the last, I would say, 10 years, we published a series of papers integrating different functional modules, including LC column, including sample preparation, including enrichment on the same silicon chip. Essentially, this is a platform technology. We show robustness, throughput, sensitivity, as well as reduced cost. So in parallel, we use the technology and develop a variety of applications to enable precision medicine. For example, we publish diabetes monitoring using top-down proteomics. We monitor environmental toxins in a drop of blood. We also study drug mechanism action through protein interactions with small molecules. We also study chemical toxicity using stem cells. Essentially, it's multi-omics. Finally, let me give you this slide. I hope I demonstrate in this webinar our NeoMix M3 emitter enabled microflow LC nanospray MS. We could achieve high flow, high sensitivity, high robustness. Also, it's plug and play. I hope it will really motivate your mass spec performance. Let me put my CEO hat on and give you this slide. So if you want to try our product, we have a quote page on our website. We now are selling starter kit for you to get started with thermal instruments. We also sell consumables as an emitter itself. Thank you so much. I think this is my last slide. If you have any questions, I'll be happy to answer. If you have any suggestions about our future webinar, We'll be happy to hear them. You can download a lot of information, flyers, specification on our website. In particular, we have 4 application notes right now. Thank you so much. I'm happy to answer any questions. Okay, thanks. So now take over.
Hello. Yes. Hi. Thank you, Dao-Jing, for the nice presentation. I hope everyone here enjoyed Dao-Jing's presentation. We really hope that, thank you for your support, and I hope that our emitter could really contribute to improving that assay performance and help you to gain better results in your research. So now we're moving to the question and answer session. So I have been collecting, there's a lot of questions actually coming through the chat. So I'm putting them together. Well, at the meantime, I would like to ask everybody here, if you have a question you would like to ask online, please feel free at this moment. So let's have some, live discussion first, then we'll move to the chat questions. All right, everyone.
So sorry, there was a question during my presentation. Sorry to cut you off. Can somebody repeat the question?
Yeah, thank you. Let's give a. All right, while you guys are thinking about questions, maybe what I can do is I move into the chat box questions so we can start a discussion. But if you think of anything, feel free to speak live, all right? So the first question we received here is, does M3 emitter work for SciEx MS instrument? So Dao-Jing, you're the best one to speak for this.
Okay, good question. We have been trying to interface our emitters with SciEx instrument. We did collaborate with our customers, Amgen, as well as Catalan. We have a prototype interface. We already talked to SciEx scientists. We're gonna initially, our plan was to start the testing with their R&D team in Redwood City this month, but given the situation, probably it'll be pushed down. What it is we want to work with SciEx to make sure everything works right, but we do have the prototypes ready. I hope this answers your question. Okay.
All right. All right. All right. So we are diligently working with SciEx right now. So we understand now after you have the SciEx instrument for quantitation, also for a lot of omics studies, in addition to the native MS. So we're working hard on that, and you'll have good news coming very soon. The second question here is, as you increase the sensitivity by 5 to 10 times, do you have a signal saturation issue?
Yes, good.
I would.
Mm-hmm.
Yeah, go ahead, I think. Yeah.
Yeah. So, good question. As I mentioned in one of the slides, so the flow splitting will increase ionization of the signal, but also reduce the background because inside smaller droplet, you have less concentrate background signals. So if you saw the saturation of your signal, one suggestion is you could inject less samples because signal to noise ratio can be still pretty high with our flow splitting. Then, of course, we also will have different kinds of angles you can go in. You can deliberately reduce your signal if you are worried about the saturation of your mass spec signal. I think the more direct way is to reduce the injection amount. I hope that will be one of the solutions. On the other hand, if you cannot reduce your IgG or other protein amounts, you can do flow splitting. That's the third way I suggest to use our emitter. In that case, you can reduce both the injection amount as well as the signal altogether, while maintaining your mass spec cleanness. As I said, a lot of times you don't want to have a lot of mass spec injected into a lot of proteins injected onto your mass spec. You can use a flow splitting, let them go to the waste or go to UV waste. Yeah. Okay.
Yeah. Can I ask a question online?
Yes.
Yes,
go
please.
ahead.
Go ahead.
Hi, it's a very good talk. This is Lindsay from Boehringer. I have a question. So for your presentation focus on native aspect, which means your mobile is aqueous, so you gain about 10 times sensitivity. I'm wondering if you run like in organic buffer, organic solvent mobile phase condition, like 50-50, for example. Do you see enhanced sensitivity increase or decreased sensitivity increase? Or
yes.
That's the general question.
Yes. So good question. Yes. So for native MS, people use aqueous solution because we want to maintain a native state. We have other 3 application nodes, bottom up, proteomics, targeted proteomics, and lipidomics. They all use organic solvent, acetonitrile, running gradient. We also work on really high organic solvent. For example, previously we collaborate with one collaborator to run 300 pesticides. Those are really high organic. So we saw consistent signal increase, but it's not as dramatic as aqueous because for aqueous it's hard to ionize. Once you have optimized ionization, you can see dramatic increase in sensitivity. What we saw was using organic, we can consistently beat the current technology by 3-fold on average. But depending on what you compare to, if you compare to high flow, then run microflow, definitely you can run, you can get 10 times or even 100 times sensitivity increase, depending on your molecule. Again, to answer your question, it works for high organic. It actually ionizes better, but relative increase depends on the molecules you are analyzing. I hope this answers your question.
Yes, thank you.
Okay, thanks.
All right, that's awesome. So just before we go to the next one, I'm also going to add a couple of input. Like I really like people ask, what's the increased sensitivity when you see signal saturation? I guess we all know that sensitivity increase of a platform or emitter here to help with your sensitivity. One is you can detect lower. But if you do not need to go lower, you actually have enough signal, like Daojing said, you can inject less, you can dilute more. In a way, you're not only saving your sample, you're also improve the robustness of the assay because you introducing less matrix into the instrument. All right, so we're moving on to the next question. It's actually a comment. My own experience shows that M3 emitter has an angle with the MS orifice in contrast to many competitor source, nano or micro ESI source, for example. Because of this angle, the orifice is much less easily clogged by neutrals. So basically, I interpret this as an enhancement of robustness. All right, nice comment, thank you. So we're moving on to the next.
Yes, I add something to this comment. Yeah, thank you so much for the comment. It's a very good observation. The main reason why right now we have angle is because we are running microflow. So for nanospray sources, typically they use a straight direct 100 degrees in or 0 degrees in without any angle because the droplet will go inside. But if you run microflow with desolvation gas, we want to make sure the ion cone is not contaminated so much. And also with IgG, so much protein gonna go inside. If we let it go outside of ion cone, we'll maintain the cleanness of the mass spec. That also enhance the robustness of the whole workflow. Yeah, thank you for your comment. I just want to add something. Yeah. Okay, now.
Awesome. Great. Yep. So the next question is, why metabolomics is not yet a target market for M3 emitters? So I interpret this as Are we going to apply this to metabolomics market?
Okay, good question. So we want to use M3 emitter for multi-omics. Multi-omics meaning proteomics, metabolomics, even nucleic acid analysis. Metabolomics, we already try. We work with thermal team last year. We did substantial experiments. It really depending on what molecules you are looking at. Because one of the challenges or one of the advantages of our emitter, but turns out to be a little bit not optimal for metabolomics, is you enhance the ionization efficiency for everything, although you did reduce the matrix effect. So in this case, for metabolomics, you are looking at the same mass window below 500 mass to charge with the solvent together. So we didn't see dramatic increase. Typically, we saw twofold increase in sensitivity for metabolites. But if you want to compare with high flow acid source, we saw dramatic increase for small molecules. I already mentioned we work with a collaborator on pesticides. Sometimes we saw 100 times increase in sensitivity. So really depending on what you are looking for. I mean, if you define metabolomics more broadly, our third application note is on lipidomics. So it's part of the metabolomics. I mean, we can talk offline what kind of specific projects in metabolomics you can benefit from our emitters. Yeah. Thank you for the question.
Yeah, thank you, Dao-Jing, very much. So I think everybody who are doing metabolomics research obviously would like to benefit from our emitter too. So like Dao-Jing said, it really depends on the compounds, it depends on your assay. So I know a lot of people right now moving from the high flow metabolomics to microflow metabolomics. In this case, this microflow platform together with our emitter is definitely going to help you to boost your sensitivity performance. We would like to discuss more with you if you have any specific applications. All right. Moving on to the next question. Will a new thermonuomics product source, ion source, be available on the market soon?
Okay.
People are looking forward to it.
Okay. Since you are listening to our webinar, I just want to tell you within this group, we are right now actively working on our own interface to thermal instruments. We already have the prototype manufacturer. We're gonna do alpha testing as soon as this week. So for those of you, if you don't have nanoflash source or nanoflash NG source, we may be able to help you interface our M3 emitter to thermal mass spectrometer, hopefully by the end of Q2. But we are also working with thermal to make sure our interface works well with their mass spectrometers without any safety issues or those kind of things. Yeah. But to answer your question, yes, we are actually working actively on two sets of thermal instruments. One is the NG source for their newer generation mass spectrometers. The other is a legacy interface, nanoflash source for older instruments like QE series. For their newer instruments like Quantiva, Explorers, and Lumos, they use a bigger ion cone. So we also are working on that set of instruments. So we'll fit almost all thermal mass spectrometers, hopefully in the coming quarter. Yeah. Thank you for that question.
Great. Thank you, Dao-Jing. Okay, the next one is pretty much based on the product and technology. I think it'd be Dao-Jing would like to answer that. In the image, I see central, this middle 8 out of the 10 emitters appear to spray LC flow. Why is that?
Okay.
We asked what's this Maybe we can go to the picture to explain.
Let me. Is this the picture? Okay. So good observation. It took us 3 years actually to finalize this design. If you look at these 10 nozzles, you probably realize the first one, number 10, they didn't spray because we deliberately blocked them. Then the internal 8, we make it hyperbolic so that this design was based on a lot of modeling, electric field modeling, as well as testing, alpha testing, beta testing. If you think about multi-nozzle emitter, the biggest challenge is the droplet. So we optimize the design as well as the flow rate for different ID. For example, we probably will launch a new product with a 20 micron ID or 5 nozzle for bigger protein analysis for native spray. So those are a different set of products. I mean, for each kinds of molecules for different applications, we have to go to the drawing board and really redesign our emitter. This is a micro scale silicon-based chip. It's not easy to do. Took us a long time. I mentioned 2007, I published the first analytical chemistry paper. Although we didn't actively working on the product for the first several years, but it did take us several years to really optimize the design, get the flow running as expected so essentially the design will optimize ionization without causing the droplet issue. So I hope this answered the question. Did I answer the question?
I think it's very clear. And what do you think? If you have any?
Yeah, did I answer the question? Maybe.
I think that's a yes. So if you have any follow-up, please let us know.
Yeah, or you can turn on your. Yeah, go ahead.
Yeah, the audio. Yeah. We're actually getting a lot of questions. I'm liking this. You know, even that we are kind of about 5 minutes towards the end, but I will try to get all the questions. So the next question, I'll combine this too, because they're both asking the robustness performance. The first one is, how long does the emitter last? The second one, pretty similar, how durable it is. How about the maintenance and how often it needs to be replaced? Yes. So I guess it could be application dependent. Xiaojing, I would like to address that based on the experiments that you have experienced.
Okay. Yeah, as Na just mentioned, it's really depending on the molecules, on the experiments. So for our application notes 1 and 2, because they focus on peptides, bottom-up proteomics, those kind of samples tend to be clean, and also you use organic solvent in the mobile phase. So our criteria is 300 runs, typically lasting 4 to 5 days, depending on your LC-MS gradient. Some people run 20 minutes gradient, some people run 1 hour, but for microflow, we don't suggest you run really long gradient. That defeat the throughput. So for those kind of samples, I would say 300 runs with 4 days lifetime. For native spray, which is the focus today, we have been demonstrating that we can run 150 runs. Those are LC-MS of 15 to 20 minutes long. So essentially, it's a 3-day experiment. This, I think, is very reproducible from end, and also it is very hard to do. If you imagine the aqueous phase with high salts, we can still run 150 runs. That's why our customer from Regeneron mentioned that we have high robustness. So I hope this answers your question. I mean, really depends on the molecules and the solvents and how much protein you're going to inject into your column, it is very robust. That's one of the main advantages of our emitters. I hope this answers those two questions.
All right. Yeah, we're actually also working diligently with our current customer because they have different applications. They're interested to see the robustness and how long it lasts. So far, we're getting good feedback. But once we have more data, in addition to our application notes, more applications, different matrix, different compounds, we'll always keep you guys posted. All right. Next question. Does the high buffer concentration used for SEC require frequent cleaning of the instrument? This is must be relevant to this native MS assay.
Yeah.
And how do you clean it if found clogged? Yeah.
Right. Okay. Very, very relevant question. Right now we have a QE plus. So one scientist just asked the question why we have an angle, right? So that's one of the advantages of using our emitter. Right now we are using 30-degree angle going in. So a lot of backgrounds, salt, as well as denatured protein, they're gonna be on the outside of ion cone. However, for people doing native MS, we know mass spec from the transfer tube and in gonna be contaminated with native MS protein after it's ionized. So in our case, we, depending on the injection amount, if you run 1 microgram of IgG, you may have to clean your mass spectrometer more frequently. This I have to ask one of the scientists in the lab. I don't know whether Peng is here. How often do we clean the mass spectrometer? My understanding is a month, depending on the injection amount. But I don't have the exact number in my head. I don't know, is Peng online? Can you answer the question?
Okay, yes. I mean, that really depends on the mass spec. So for our QE plus, normally, I guess you mean cleaning the ion transfer tube, not the essence. So for ion transfer tube, if we do native spray, we normally clean like once in 2 weeks.
Also depending on how many samples you run, right? You run consecutively unstopped. Yeah. Okay, I hope this answers your question. I mean, essentially it's depending on the mass spectrometer, QE plus, HF or Lumos and UHMR, or you use Q-top instrument, maybe that's a different story. But in our hand for QE plus, my understanding is typically a couple of weeks to a month, depending on the injection amount of the IgG antibody. Yeah, if you do native MS. Also depending on the salt concentration. If you run really high salt, I think 200 millimole is really high already and maintain most of native state. I think it's good enough. So to answer your question, a month, roughly, yeah. Okay, now I hope this answered the question, this question.
Yep.
Okay.
I think so. We're always welcome for further discussions, guys. And this is great questions. The last two questions, we're going to borrow a couple minutes from you. The second to last, I heard that you guys have a smaller than 5 micrometer emitter under development that could be useful for the nano LC scale assays for limited amount of samples, such as single cell proteomics. When will that, is that right? When will that be available?
Okay, just to lead the audience, we are testing our nanoflow solutions right now. We already make the smaller ID, also different geometry, M3 emitters. If you remember, my first publication in analytical chemistry in 2007 was for nanoflow applications for our emitters. But these days, we want to increase the robustness while dramatically increase the sensitivity. So we are exploring different kinds of designs, geometry. In the beginning, we thought microflow LC, nanospray, or mass gonna be our main application. As we talk to customers, we realize nanoflow right now is another killer application because, as you mentioned, that a lot of people are interested in single cell analysis. A lot of people are interested in one drop of blood for multi-omics analysis. That actually give us the incentive to further develop our nanoflow solution. I can't give you exact timeline, but what I can say is we are actively working on it. We already made the preliminary designs. We are testing those. If the performance is good, we definitely will launch this product for nanoflow applications. Okay, now next question.
Sure. Great. All right, the last and also final question is about samples in compounds. So what approximate fold increase in sensitivity for oligonucleotides? And what about other protein complexes besides antibodies that you have studied using the emitter?
Yes. So thank you for the question. As I mentioned previously, our emitter can be used for any molecules that can be analyzed by mass spec. We are a small startup company, so we didn't have the manpower resources to test all the applications. However, our customers have been using our emitter for nucleic acid analysis. For example, Regeneron, they actually try our emitters for nucleic acid analysis. I don't know what kind of experimental conditions, what kind of fold changes they observe, but seems to me it works. And we are also working with other customers on different type of molecules. We previously have shown our emitter work well for glycans. We are working on glycomics application notes pretty soon. So whatever molecules that can be analyzed by mass spec, I think our M3 emitter will help you in terms of sensitivity, robustness, and throughput. But again, it depends on your experiments. So we will be very happy to talk to you. You can just contact us. We have emails in the email list, or you can contact Stephanie, ask the question. We can follow up with our customer support team. We always want to talk to the customers to help you address your challenges. Yeah. Okay.
All right. Thank you, Daojing. The last final question, I think, will come down to the pricing. Can you give a rough price for the M3 emitter? So I guess here, you know, we have that in the product flyer. So we're, like Daojing said, we're a young emerging company with a new technology. So you can find this information. You know, product portfolio is small, but it's very focused. So the emitter sells for $299, single emitter. And we have, if you want to utilize the emitter, there's a need of a starter kit that is list priced at $39.50. For more information, quotation, and pricing, please feel free to reach out to the quote link and the NeoOmics sales and marketing team. Yeah. All right.
So over here,
thank you.
Yeah. Yeah.
Right.
Okay. Thank you, everybody, for your interest. As I said, now Stephanie and myself are happy to receive any email questions. We'll address them. If you are interested in our product, go to our website, download our application notes. Thank you so much. I hope everybody stays safe and healthy during this difficult time. Thank you, Na.
Right. Thank you, Daojing. Well, once again, from the NeoOmics team, we want to appreciate your time to join us in this difficult time. Stay safe, stay healthy. We'll keep learning. Thank you. Bye.
Thank you. Bye.